Okay, so maybe we can start the seminar. So I will just briefly mention that to the second seminar of the series on the mathematical laws of morphology and biomechanics. We have also working groups. I welcome everybody to participate to the working groups as well. They are held quite regularly. So we had one last week, I think, and we'll keep having new ones in the coming weeks. And well, today we have one of the speakers, that Katrina is going to introduce in 1 second.
Hi, everyone. It is my very great pleasure to introduce Madhu, who is an associate professor of mechanical engineering and materials science at Yale University. He received his PhD from Cornell, and he studies the mechanics and control of how animals move. Some of the research projects he's currently pursuing include locomotion in animals, the geometry of joints, the topology of shape-changing skins, and the rheology of muscle. Motivations and applications for his work include biomedical sciences, evolutionary biology, robotics, and just plain curiosity about everyday observations. So with that, please take it away, Madhu. We're very much looking forward to this.
Thank you, Kathleen, and thanks for everyone for joining in. I'm cognizant of the challenges of virtual seminars, so I'm delighted to have this opportunity, though, despite that, and the fact that I did not have to deal with airports is the icing on the cake. We just make sure that you are able to see.
It looks like we're seeing your own
the same
screen.
Yeah, got it. It's just once more, and I will get it right this time. You're hopefully now seeing my
excellent
a big foot.
Yes,
in the sand. Okay, I invite questions all along the way. Please you don't have to wait to the end of my talk. I'm going to try my best to reach the end of my talk in 5 minutes, and then present a lot of detailed evidence on the basis of which I'm going to make all the claims I'm going to make. But let me first introduce my lab as a way of acknowledgement of people who fuel my ability to think. There's a gap in the middle, and the gap just indicates that these two just defended and have more than 1 foot out the door. Nilima and Koi are starting postdoc positions in different places. My current group is these four, and this is by way of also making a plug that we have openings for PhD students and postdocs in my group. Our work spans multiple topics, very generous funding from multiple sources makes these things possible, and here I list the main funding sources that have generally funded my lab, not just the work I'm presenting today, and I thank them for that too. Okay, that said, let's dive into today's topic. Almost everything I'm presenting today are in 2 papers. There are additional pieces and background material that are in these other 2. That's a book chapter, that's a preprint of a paper that's in revisions. All of those PDFs are available. If you flash your phone at that QR code, it will take you to my lab webpage, and they're available on my lab webpage. The work I'm presenting today is almost entirely funded by the Human Frontier Science Program, and I acknowledge their generous support of ideas good and crazy, and we fall in the crazy bin, I think. So that said, let me tell you about today's work, most of which was done by Ali, who's a PhD student, and Carolyn, who was a postdoc in my group. Really brave undertaking of experiments and calculations that I wish to acknowledge their contributions. A lot of my own thinking has been quite heavily influenced and shaped by these 3 people. Shreyas and Mahesh also were co-authors on the foot and fin work that I'm presenting. Daniel Lieberman, with whom I did a postdoc, has been a good critical voice of helping me think about evolution and mechanics. These are people from Yale who helped us gain access to fossil materials that was useful and used in our study. Today's talk, I'm going to talk to you about feet and arches. Much of the talk, I'm going to talk to you about feet. At that point, it'll probably become obvious to you what I'm going to say about fins, and nothing more needs to be said about fins at that point, I hope. That's the intention of how I'm structuring the talk. And the main result of the talk is the following. When you look at human feet, you see in many feet a big pronounced arch. In some feet, you do not see that arch. But we also have a second arch shape of the foot in the middle of the foot running across the width of the foot. And the long-standing question has been, why are human feet so stiff compared to, say, other primates? And they're stiff. Well, what does stiffness help you? It helps you push off on the ground using the ball of your foot, a style of walking most of us use. We tend to strike on our heel when we walk, for example, and push with our front foot. When we run, people use many different styles, but still we push off with the front of the foot. And when you're doing that, you don't want your foot to be flexible like this thin sheet of paper, because the lightest load and the whole sheet just gives, and then you're no longer going to be able to push with the front of your foot. So you want a stiff foot. And what I'm claiming, and that's the talk really, and that's the conclusion of the talk, is that it's this curvature along the width, the transverse direction. It's the pizza eating strategy or dollar bill strategy to go when you go to the vending machine, where you slightly curl a thin elastic sheet, and that makes it vastly stiffer. And towards the end, I'm also going to show you how a related phenomenon probably appears in fish fins, but that's ongoing. Part of it is ongoing work in my group. So that's the talk. The talk is transverse curvature in feet and fins has a big responsibility for what makes them stiff structures. And we care about stiffness from point of view of propulsion and being able to withstand the loads that are required for propulsion. The rest of the talk is to try to present you the evidence and way by which we arrived at this statement. Let me start with giving you a sense of the numbers and scale in human feet first. So here is a person running on a treadmill. While they're running on the treadmill, we're also recording ground reaction forces, and the trace you're seeing is the vertical component of the force. And you see that when a person's pushing off, they're experiencing many times their body weight. You'll see that number go above twofold. Every single step your foot deals with 1 to 5 times body weight, depending on the speed at which you're walking or running. And when the foot is experiencing that body weight, mostly you're supporting the weight with the front of the foot. And these dot patterns are here not for measurement sake, it is for illustrative purposes in this video. And you will see the middle of the foot barely bent. There is some amount of flattening of the arch. It looks like the entire structure is bending under the load a little bit, but just ever so slightly. It's remarkable how stiff the foot is when you think about the loads. It's not just in running, as I said. In walking, it's very similar. You don't experience 2 times body weight, but each foot experiences about 1 time body weight when you're pushing off with the front of the foot. And while you're doing that, simultaneously towards the heel, you have rest of the body pushing down at your ankle, and you have things like the Achilles tendon pulling up and providing a couple. The net effect of this couple is your ankle becomes relatively stiff, and most of the deformation just when you're pushing off is happening within the foot, and that's when the foot is experiencing all these very large bending torques, and it has to be able to withstand it. That topic is of interest not purely from question of can you push yourself by using your foot as a lever, but it's also a question of how does it affect the energetics of the foot and therefore of all of your locomotion. Here's an experiment, a very clever experiment done by Takahashi and others. They were measuring the deformation power associated with the foot as a function of the stride fraction, meaning this is when your foot touches down and that's when your foot lifts off. The deformation power they're plotting is the mechanical power associated or rate of change of energy associated with that bending motion that I'm showing you. There are many curves, but all of the curves follow the broad trend. There's some amount of work absorption in the foot. Then for most of stance, very little is happening in the foot. And just before you're about to push off, when you start loading the front of the foot, is when you see the foot absorb a lot of energy. So there's a lot of negative power and return some fraction of it as positive energy just when you push off. The amount of negative work done on the foot, how much you absorb, is of course a function of the stiffness of the foot. And in this experiment, they had people wearing a very flexible shoe and provided a stiff carbon fiber insole to increase the stiffness systematically. And very not surprisingly, you see that the stiffer the foot, the lesser you absorb. Somewhat maybe surprisingly, you also see it return more energy back when the foot is stiffer. The story how this affects whole body energetics is a lot more complicated. The short version of an example of the complication is stiffer is not necessarily more energetically efficient. The least energy consumption in walking appears somewhere in this intermediate stiffness. That's to point out that stiffness is a variable that has measurable impact on the performance of the foot. It has measurable impact on performance of the whole body energetics, and you care about the stiffness being approximately where the human foot stiffness is. Okay, great. So we then should by now clearly understand, its structure is in front of you. We should understand what makes the foot stiff, and we do understand a lot of it. The work in this dates back at least 100 years, but I would like to bring to your attention one very nice experiment done in the 80s, which is not using live feet. This is using amputated feet, so feet of people who had their foot amputated, and that they donated for science. The foot was amputated for other reasons, of course, and they're then testing the stiffness of this structure as a mechanical structure, and the interest is to try and mimic this type of loading. So I want you to pay a little more attention to this experiment because I'm going to come back to this type of experiment in the later in the talk. What they're doing is they've taken this transected foot, attached it to a mechanical system that displaces it up or down, and the whole foot is setting such that the front of the foot is on this trolley and the heel is also on a trolley. That's because as you compress the foot is going to compress vertically and expand along its length. In order to allow the expansion freely, which is what would happen when you're on the ground, pushing with your toes, they mimic that free expansion using these low friction sliders. But remember, I told you that the ankle needs to be relatively rigid, which means you need a force couple at the ankle to support the torques applied by the front of the foot. And that force couple in this experiment, instead of the Achilles tendon pulling up, the ground is pushing up. So it's a very clever way of replacing a tendon with the surrounding mechanical boundary conditions just correctly picked. And then they measure load as the vertical load as a function of the vertical displacement. The blue curve is the intact foot. Okay. Now you look at the bottom of the foot. Remember, I told you this is big longitudinal arch, and the 100-year story has been that the longitudinal arch, together with these thick elastic tissue beneath, thick as in the sense of you can see that it's visibly thick, but it's not thick in the sense of approximating as a thick body. It's still fibers. And those fibers, called the plantar fascia, together with the longitudinal arch, behave like a bow and a string. So if I give you a bow and a string, the bow itself, because of its intrinsic material properties, has some amount of stiffness. Now, if I have a very stiff string running across the bow, it will be a lot stiffer because when I try to bend the bow, I'll have to stretch this string. And the stiffness mapping from the stiffness of the plantar fascia to the longitudinal arch becomes proportional to the square of the height of the arch. And that's why there is a lot of emphasis, and correctly so, that the height of the longitudinal arch in humans is a big factor that allows the foot to be quite stiff. And the way they tested it in this experiment is they took the intact cadaveric foot and just cut the plantar fascia. So the bow is still intact, but the plantar fascia can no longer support any loads. And you see that the load displacement curve shifts down. in the sense of becoming softer. But then they started cutting other ligaments that are inside the foot that are holding the bones together. And of course, then you see a drastic drop in stiffness because the bow itself is losing its structural integrity. The main point I wanted you to take away is how the longitudinal arch and the height of the arch impacts stiffness of the foot because of this bowstring effect, and that is a big part of our understanding. If you compare human feet with other primates, for example, there are running monkeys like the macaque and vervet monkeys. In those monkeys, you don't have a visible longitudinal arch. You still have the plantar fascia. The foot is, in fact, you can map the bones of the foot one-to-one between the monkey and humans, of course. And repeating that experiment in those monkeys, the same set of authors, in fact, found that cutting the plantar fascia essentially has little or no effect on the load displacement curve. In other words, it is the longitudinal arch that helps the plantar fascia contribute to foot stiffness. And therefore, in the absence of a longitudinal arch, the plantar fascia does nothing. Very nice, clean understanding of how this the structure is functioning. But there are all kinds of debates about it. And to understand the debates, it's helpful to know a little about what are humans in the sense of relative to other primates. Let me tell you a little about the story of human evolution so that we can understand why are we making this primate comparison in the first place. That story has many key points, but the point where I would like to start is very recent. So 7 million years ago, where the lineage leading to chimpanzees split from the lineage leading to modern day humans. Along the lineage to modern day humans, you had many species. Some of the ancestral relationships are still debated. Those I've shown as dashed lines. Solid lines, we have a lot of compelling evidence that the ancestral relationship is probably correct. But along the way, first around 4.5 to 5 or so million years ago, Ardipithecus emerges. Ardipithecus is remarkable because when you look at its pelvic adaptations and so on, it looks a lot like a bipedal animal. It doesn't have a pelvic cup which is pointed down. It has many other adaptations which make people think that it was bipedal. And by 3.5 million years ago, you have Australopithecus. Australopithecus, one very popular fossil that you can go to the Smithsonian and see, is Lucy. So this animal shares a lot of similarities with the chimpanzee skeleton, but its lower body looks a lot like a bipedal animal. In fact, very remarkable thing that has been found is this long trackway of footprints that are made by fairly surely Australopithecus, and that footprint looks like a bipedal walker. So we think that was walking, and by analogy with current day primates, the guess is that they were also probably very good at climbing in addition to walking. So sort of not like us, a little more adept at climbing, but they could sprint. I mean, your average chimpanzee can sprint bipedally quite fast. But further along, around 2 million years ago, is when genus Homo, where Homo sapiens, Homo erectus, as it's called, appeared, and these are again popular names. We know, and this is a very long literature that I'm summarizing in a very simplified way. We know that Homo erectus was probably very good at walking, but also at long distance running and launching projectiles at very high speeds. These are topics on which we've researched and have many others. What changed from here to there? Well, we look at the foot. The human foot looks quite a bit different from the Australopithecus foot. And one difference is also a topic of major debate. So let me tell you about what's changing in the foot over time in order to begin to appreciate why that longitudinal arch bowstring story is not yet the full story. This is simply a memory aid for you. What observation I want you to note is that not all human feet are as stiff. If you took a healthy person and asked them to stand and support their body weight on the front of their foot and raise their heel up, they can do that. There are some people who, for various reasons, diabetes, neuropathies, and other injuries, can end up with a very flat foot. And you ask them to raise up their heel, that's how far they can raise up their heel. And what you will see when you ask them to do that on top of a pressure mat where you're recording the contact pressures, the normal traction really is, if a person with a healthy foot walks across, they would see a very high contact pressures at the heel and high contact pressures towards the front. But someone with a flexible foot like that, who cannot lift their heel up, will also see huge contact pressures in the middle of the foot. You can also see that more clearly in this still shot of a bonobo that's walking bipedally. And as it's walking, the middle of the foot, and by middle of the foot, I mean this region in the equivalent to this region in the human foot, is bending quite severely. And that's what happens in people without arched feet. Seems to support the idea that longitudinal arches are important. On the other hand, you see a lot of people who will appear to have flat feet and have no difficulty raising their heel up. In fact, if you wind the clock back and go to the evolutionary story, Australopithecus afarensis, for example, we think walked bipedally, and this is the footprint, preserved footprints I was referring to. It shows an adult and a juvenile walking next to each other. It's about a 14-meter long trackway, beautifully preserved with a deep heel imprint and a deep forefoot imprint. So it's a remarkably human-looking gait, but the feet of Australopithecus that we can find don't appear to have much of a longitudinal arch at all. It doesn't have a longitudinal arch, could walk human-like. There are living humans who don't have a very visible longitudinal arch and can still lift their heel up. All sorts of debates start coming up if you then look at the surgical literature, where in some people with collapsed arches, the arch is reconstructed. Sometimes it leads to big improvements, other times it has no effect in terms of their ability to lift their heel up. So what's going on? There is a lot of debate, but the debate is pretty much entirely surrounding the longitudinal arch, and the pizza curling effect has really not been investigated in any of these feet, not let alone human feet. And that's the story I'm going to tell you about what we did. Is the loading picture and so on clear? If something is not, please again stop and ask questions. I'm happy to take questions along the way. Okay, so here are the first few things we did to try to understand why when you curl a dollar bill it becomes so much stiffer. That fundamental result is, I'm going to show you our experiments, but the real attribution to that result should go back to Gauss. It's Gauss's theorem of equivalent, which is a remarkable theorem as it's called, which talks about how curved structures, depending on the type of curvature you have, will lead to stretching thin curved elastic sheets. Sorry, I think I heard someone ask a question. Okay. So maybe there's some microphone that's on and I can hear their background sound. All right. So let me then tell you about curved elastic sheets. First, think of a very simplified setting. I have a wall, and to the wall, I attach a thin elastic sheet that is slightly curved along its width. And I'm going to try to displace this edge and measure the reaction force upon displacing. So how much effort do I take to displace a cantilever that's slightly curved? This also should look like your measuring tape, engineering measuring tape, for example. All of those have this type of shape. So we did numerics with finite element simulations. We did physical experiments with PDMS shells that are at least visually don't look like thin, shallow shells. They're pretty fat and they're virtually semicircles, so they're very highly arched and very thick. And in those experiments, we clamped one end and applied an edge load to mimic this type of loading to ask the question of how stiff is the structure. And here I'm plotting stiffness as a function of transverse curvature. The circles are the numerics, the diamonds are these PDMS shells, and the stars are the PDMS shells, but without transverse curvature and only longitudinal curvature. I don't have anything like the plantar fascia here, but I have longitudinal curvature. Broadly speaking, as I increase curvature, you see stiffness increase, but a lot of scatter. This is a log-log axis. That's a lot of spread. So there are other things affecting the stiffness, not a big surprise because we varied the thickness, the length, the width. We're changing other things too, and they also affect stiffness in the dimensional sense. But feet are not these continuum shells. So let's try to add a little bit more complexity before going to the real feet. And the complication I'm going to add is think of a discrete structure of bones, 3 bones, that are arranged in a transversely arced way. By that I mean that the bending axis, there's a hinge at the base, and that hinge axis is slightly offset as you go from one of those long bones to the other. Each bone is attached to the wall with a spring using the arrangement that is shown here. Right, so there's a spring, that's the hinge, and so if I try to rotate this hinge, I will stretch that spring, so it's like some intrinsic stiffness for this structure. Okay, and these 3 are also interconnected by springs at the distal end. This distal end springs are going to stretch only if there is any stretching within the plane of the structure, not bending outside of the structure, unless these hinged axes being mutually misaligned means that as I push these rays up, they're going to try to splay apart. I mean, if my video is visible to everyone, let me do an animated demo. Right? Think of 2 of those bones with springs in between, and let's say the hinge axis is such that they're parallel, and therefore if I try to displace it out of plane, nothing interesting is happening. I'll just measure 2 times the stiffness. But if the hinge axes are misaligned, as I try to push it out of plane, I will also stretch the spring in between them. So that's coupling bending to stretching. That's really what underlies why curvature made this stiffer, and a similar phenomenon would manifest here. And in experiments with feet of different thickness, meaning the height, how far this spring is away from here, different length and different width, you see, broadly speaking, an increase in curvature leads to an increase in stiffness with quite a bit of scatter. So what we're going to try to do is take this understanding of curvature-induced bending, stretching, coupling, and try to derive normalizations for measures of stiffness and curvature so that we can compare structures of different overall dimensions on a single non-dimensional plot. So that's what I'm going to do. So how do we find that normalization? Well, I am going to show you more details, but the underlying intuition is the following. If I look at a curved structure head on, so I look at the curved dollar bill head on and try to displace all its material points normal to its surface, I have to grow its perimeter, which means bending will cause it to stretch, and the amount of stretch depends on the curvature. The more the curvature, more the stretch. And that is what leads to curvature-induced bending, stretching, coupling. And the non-dimensional variables that come out as being important are the stiffness of the structure relative to another, which is identical in all regards other than the curvature. So how much is the curvature alone leading to a stiffness increase? That non-dimensional ratio of stiffnesses isolates the contribution of curvature. And it's not curvature itself that matters, but curvature times length squared by thickness comes up as a non-dimensional parameter that would govern the non-dimensional stiffness. So let's try to understand this a little bit more. Where do all these things come from? So one way to understand it is numerics, and I'm going to use numerical demonstrations instead of just analysis of the underlying equations. Okay, so this is the loading setting. So I'm going to try to displace the tip, measure the reaction force. One way of maybe thinking about the whole thing is, well, I have a beam whose neutral plane is away from the material surface. It's the classic I-beam. And we know that the bending stiffness of a linear elastic beam depends on this second moment of the cross-sectional area, which I can increase by simply curving it. In other words, material points above and below the neutral plane are going to be in compression and stretching, and the amount of compression and stretching depends on the height of the neutral plane away from the material body. So maybe that's all is happening. The first hint that that's not the case comes up when you apply a total displacement on one edge, hold the other edge clamped, and look at the distribution of displacements. Almost all the out of plane displacement is concentrated towards the tip. In the linear beam, what you'd have seen is a gradual increase in the displacement. So you would see displacement go all the way to the base. Here you see something happening at the tip. And in fact, if you plot this, one of the stress components associated with stretching or compression along this direction, so that's sigma xx, where x is the long axis, the neutral plane hypothesis would predict that the sigma xx equal to 0, no compression or no stretching happens exactly at the neutral plane. That's the dashed green line on this plot. The black line shows the actual contour line corresponding to no compression or stretching. And what you see is towards the tip, you have a departure from the linear beam theory. And that departure is happening in a boundary layer, which we would like to understand where that boundary layer comes from. That boundary layer comes from a competition between bending and stretching. Bending energies go like thickness cubed, but stretching energy goes like the extra strain introduced due to the change in stretch, which is because of the curvature. So the total energy that would be minimized, which is how the elastic structure would respond, happens when these two become balanced with each other. You're not bending everywhere or you're not stretching everywhere. And that leads to a scaling for this boundary layer. And the comparison of that boundary layer to the total length is the dimensionless parameter, which I'm calling normalized curvature. Okay, and that normalized curvature is this quantity. So let's go back and plot the same data in terms of these dimensionless variables, where I'm going to look at the ratio of the actual stiffness relative to a hypothetical flat structure as a function of this dimensionless curvature. So these exact same data here, this is a physical experiment. So I can build a hypothetical flat structure. What you see is normalized stiffness is pretty much entirely governed by this normalized curvature. We are not in the shallow limit, we are not in the thin limit, and yet the theory is fairly robust. For this one, we can write down the governing equations and tell you how exactly to collapse the data. That's kind of boring. That's a homework problem. What we're trying to ask is, will this continuum theory, which strictly does not apply to this discrete structure, is it still a robust theory in terms of capturing the principle? And what we see is that same dimensionless measure of curvature is able to collapse the discrete structure data as well, giving us more confidence that this is the right curvature to be using when comparing feet of different sizes or structures of different sizes. Okay, I'm going to now ask the question, great, I could do all this here because I could normalize my stiffness. I spent all my effort explaining how to derive this normalized curvature, but the real experimental difficulty is normalizing that stiffness, because I cannot build that foot, which is identical in all regards but for curvature. That foot does not exist. So how do I, in a real foot experiment, ever perform something like this normalization? That is the real question. In other words, how do I know how much did transverse curvature stiffen my structure? And here I'm going to show you an experiment that Ali did as a short video, and I will say a few words here and there when that video plays. And the idea here I want you to keep in mind is what curvature is doing is coupling bending with stretching. So what Ali did was build a structure that is two of the bones with their hinges parallel to each other. But if you now induce curvature, which causes the hinges to become misaligned, the whole structure became stiff and the weight got lifted. However, if you cut the interleaving elastic material, in this case just a sheet of paper, that causes the stiffness to collapse as if the two metatarsals, those two bones, are acting independently. That's the key, simple but key idea of how we can hope to test this in real feet. So let's make sure that this video has some sanity beyond just the video. So you can measure the stiffness of a curved mechanical structure where I've cut these springs, or you can measure the stiffness of a flat mechanical structure where these hinges are aligned, the resistance or absence in the spring makes no difference, and find whether these correlate. This is just an experimental examination of that same idea, and you see a near perfect correlation between cutting the springs and flattening the ash. So that idea carries over in these discrete mechanical structures. So cutting those transverse springs effectively disrupts the function of the transverse action. We can do this in human feet. That's the idea. And we're going to do this in human feet following the experimental approach that I showed you earlier. We can mimic the type of loading you see in this material testing arrangement. These are called 3-point bending tests because 1, 2, 3. Unlike Kerr's experiment, which were done with amputees, we did our experiments with cadaveric human feet. So people who die, donate their body to research. Those feet are preserved at -80 very promptly. So they are called fresh frozen cadavers. We thaw them and conduct these experiments using a material tester. So to remind you, the effect of cutting the plantar fascia was a 25% or so decrease in the energy stored associated with this load displacement curve. By comparison, those monkeys, approximately 0%. That's the reason we think that the plantar fascia is so important in human feet. That's a pretty big increase in stiffness compared to flat-footed monkeys. We did the same experiment, but instead of cutting the plantar fascia, we cut the tissues in between these long bones, like that video with the plastic model. Like we cut Ali cut the plastic, the paper sheet holding the two plastic bones. Here we're going to remove the elastic soft tissue in between these long bones and conduct the same experiment. And when you do that, the intact load displacement curve, that's load, that's displacement, goes down quite a bit. And in 2 feet that I'm reporting here, it goes down by about 50%. That to me is a pretty big increase, and it was dramatic, surprising, all of those adjectives. But we also know something else we can do, which is estimate that dimensionless curvature. So we went ahead and estimated dimensionless curvature for these 2 feet, but we did this for lots more feet. But before I show you what the dimensionless curvature in human feet look like, I want to also point out that this is not true just in cadaveric feet. We can do this in live humans, but not exactly the same. You can't go around cutting tissues in live human feet. However, and here again, Ali had a very clever idea. He said, well, I can't cut and reduce stiffness, but I can add and increase stiffness. I can add stiffness by using an elastic tape I can wrap around a foot. So for example, in this mechanical model, here is a curved, here is a flat model where the axes are hinged. Here is a curved model where the axes are staggered, so their axes are not exactly parallel. But if he adds a rubber band around their tip, essentially increasing the transverse stiffness, the structure becomes stiffer. He did the same thing in people who are now all marked up and walking on a treadmill, either walking freely or walking with a tape that's very loosely applied. So it gives you the sensory experience of a sticky kinesthetic tape applied to you, but not the stiffness due to the tape, or the tape applied pretty tightly using a consistent protocol. And what you see, these are the power curves like I showed you with Takahashi's experiment, is again, the midfoot absorbs a lot of power just before push-off, and during push-off returns a lot of the power. The stiffer structure should absorb lesser and return lesser, and that's roughly what we see. So the negative work done by the stiff foot is consistently lesser than a free foot. Stiff as in hypothesized stiff, the one with the tape, and also returns lesser. So overall, the taped foot is not deforming as much in any direction. You can also look at measures of stiffness by just plotting the midfoot bending torques as a function of the midfoot bending angle and estimating this is the loading and that's the unloading curve. You can estimate what is the stiffness of this structure. And you again see that the stiffness changes systematically depending on whether you have a tape or not. And it's not just the sensory experience or all the muscles in your foot and so on don't change what the effect of transverse stiffness on longitudinal bending stiffness of the foot. So you have this cool cross-axis coupling going on. Okay, so now I would say I'm fairly confident that this is the principle operating, and based on these toys, I think CL squared over T, that dimensionless curvature, is the thing I care about in discrete structures. I want to be able to measure this, and not just in live human feet, but let's first see how to measure it in live human feet. I can look at a CT scan of a live human foot, look at these metatarsals arranged in a transverse arch, draw a curve on top of it or through the middle axis, your favorite pick, and estimate what is the mean curvature. It's not a constant circle, so curvature is different in different areas, but I can go from one end to the other and look at the angle accumulated by the normal to the curve divided by the width, and that's approximately my mean curvature. You can do this in another interesting way. That's because the front of your foot is level on the ground and the middle of your foot has this arch. So it's not your simple tube curved like I was showing with all these other models. What that leads to is identifiable bony landmarks associated with these long bones that have a twist about their long axis. I've highlighted it in this cartoon. So if you think of these as approximately rectangular ends, they're not, but you can identify specific bony landmarks to fit these. That rectangle of, say, this fourth bone undergoes a twist as you go along its length. I'm going to call that metatarsal torsion. So that torsion is really an estimate of how much the normal to the whole foot changes as you go from one end to the other. So the torsion angle divided by the width could be a candidate mean curvature. We took 6 cadaveric feet and 6 live feet, CT scanned them, and made both of these estimates of curvature. And what you find is the torsion-based curvature and the external surface-based curvature correlate reasonably well with each other. It's not perfect correlation. The slope is not 1, but one variable contains quite a bit of information about the other. That's what we are going to use because a lot of fossils are incomplete. So here are the key feet we're going to analyze. That's humans, there's chimpanzees. These three are members of the genus Homo, Homo habilis and erectus around 2 million years ago. Homo naledi, which is really cool fossil. This animal practiced ritual burial. So you have these beautiful intact fossils, and you can actually see it has a very visible transverse arch. But there are older fossils we want to look at, the early start of bipedalism. Australopithecus afarensis, Lucy, who left the footprints, or some relative of Lucy who left the footprints around 3.5 million years ago, much to our delight. The fourth metatarsal is relatively intact and available for analysis. This is an even cooler fossil. It's called the Bertelli Foot, named after the region where it was found, dated to around 3.5 million years ago. Very little has been found for this animal. So we hope to be able to even generate hypotheses about what kind of animal this was because the fourth metatarsal is available. What we did was, to remind you, we plotted we estimated the metatarsal torsion. We estimate other dimensions based on that one bone. Well, we have to use some animal template, and animals vary in the relationship between this bone geometry and the total size of the foot, the length, the width and thickness and so on. So we did a Monte Carlo approach, and for each of these species, estimated what the expected spread in that curvature parameter, CL squared over T, is going to be. I'm plotting that on a logarithmic scale. The bottom two are those running monkeys for which Kerr et al. Bennett et al. Collected the data in the 1980s. For them, they're such flat feet, even in the transverse axis, that the mean, the black dot, doesn't even show up on this logarithmic scale. The third one up is the chimpanzee, again, very, very flat. The gorilla, not so flat-footed in the transverse axis. Its longitudinal arch is pretty flat. It appears to have a consistent transverse arch. Humans appear, our dimensionless transverse curvature is between 15 and 16, pretty consistently, at least in the samples we found. All of the genus Homo around the normal human variability. Australopithecus afarensis, way more arched than the gorilla. Not quite within what I wouldn't consider this within normal human variability, but it's getting close to normal human variability. Bertelli, surprisingly, very, very much within normal human variability. So we're pretty stoked by this. We think that the afarensis was able to walk with the heel to toe style of walking because of its transverse arch. The transverse arch also has appeared in other species at the same time, not just afarensis, or around the same time. They probably appeared earlier. We don't yet have data and analysis on earlier time points. But in this sparse fossil record, it looks like a human-like transverse arch appeared at least 1.5 million years before genus Homo, and around the same time as other evidence for bipedal walking appeared. So I will end my foot portion, and you already know what I'm going to say about fish fins, or mostly know what I'm going to say about foot fins. In the human feet, I would say the transverse arch contributes significantly, but this is causing us and others to re-examine even what we mean by a flat foot disorder. Everything is currently classified based on longitudinal arch. So we really need to think about what the transverse arch is, and those are data hopefully will come in the future. But the geometry of the arch alone is not all important. As I said, it's about the bending axis of these metatarsals. You may look flat, but the preferred bending axis may be slightly different. How do we even start to predict what is the preferred bending axis of a joint based on its geometry? That's a tough question. I see Armita in the audience who has pretty deep things to say about that question. That's a very hard question that would have to be tackled as well. And that is related to how fish fins work. And can I take about 2 or 3 more minutes, Kathleen, to show what I have about fish fins? Okay.
Absolutely.
So why am I thinking about fish fins? It's because really, let's wind back the evolution clock a lot more. All vertebrates are descendants of aquatic fishes. We are fishes, in fact. So around 420 million years ago, the lineage of fishes split into things that are called ray-finned fishes. Almost every fish you find today in the ocean is a ray-finned fish. And lobe-finned fishes, there are these strange creatures called coelacanths and lungfish, they're examples. And all land vertebrates and aquatic mammals are lobe-finned fishes. So we want to understand how did movement on land first emerge from an aquatic ancestor? But the limb structure differs quite a bit between lobed and rayed fins. So what we want to really understand is how did potentially early land vertebrates use a fin-like structure on land? It's horrible to call this feet because they're really fins, but they were being used like feet, like this mud skipper video that was running there. So that's a very recent transition from water to land. It's using a ray fin structure. It's so happy moving on land, it spends over 90% of its adult life on land. So we're trying to get at those questions, but our initial baby steps towards that is to first ask, how do we understand stiffness of a whole fin, and is there something like curvature-induced stiffness? You don't only care about it on land, you also care about it in a fluid medium. Here is a very sophisticated experiment with 2 cut-off coffee cups with fins attached to it on a wooden stick, and they're spinning because I'm blowing air on one half of it with a handheld vacuum cleaner rigged up to become a blower. And that interaction with the flowing air is much stronger in the coffee cup with the curved fin because it's a stiffer fin and therefore it's spinning faster. When you have very flexible fin, you do not interact that much with your surrounding fluid. And that's the motivation to think about whether curvature in fins can work. The principles are related but a little different because the fin is made of these rays, that are themselves quite flexible and have membranes in between, which would act like the tissue in between the things. So it's maybe instructive to think of a two-ray unit cell like my demo, but now think of my arms themselves being more flexible and not a very rigid. In that system, you can go through the governing equations and realize there's really a competition of three things going on. The ray likes to bend in its soft direction if the ray was not exactly circular. The general ray, which is not perfectly symmetric, has a preferred bending direction, has a stiff bending direction, and if those are not perfectly aligned with the membrane, depending on how the ray deforms, it will stretch the membrane. The competition between these three leads to one length scale, which is the bending stretching competition. That length scale now depends on the anisotropy of individual rays, the systematic inhomogeneity between adjacent rays, and how stiff the membrane is. And that's one parameter which governs this bending stretching coupling again. But fins are cool because I can think of a curved fin with anisotropic rays and therefore this bending stretching coupling, but I can also think of a flat fin. And just because of the ray geometry, this composite structure can mimic curvature without having any visible curvature. So we got excited by this idea. So we went to the fish market, got a mackerel fin, put it between plastic sandwiches, stick it in the micro CT scanner, and quantify the second moment of the cross-sectional area in adjacent fins at different cross-sections. What you see is indeed a flat fin has systematic variation in the preferred bending direction as you go from one ray to the other extreme, but the systematic pattern itself depends on which cross-section you're looking at. We have no sane hypothesis to date, I would say, for why this pattern, why the dependence in the position and so on, but we do see that a flat mackerel fin even has embedded within it something that could mimic curvature. Whether that works, we have not done the experimental test. We've tried to use the theory to think about whether it could work in the first place. Because think about the membrane stiffness, right? They think of this as the membrane stiffness parameter, everything else being held constant. If the membrane is extremely soft, it's as if there is no membrane. If the membrane is extremely stiff, it doesn't matter what the rays want to do, the membrane will force them to bend together. There is no bending, stretching happening. There's this intermediate range where somehow curvature, meaning slight changes in curvature, can actually lead to a big change in the overall stiffness. These are dimensionless equivalents of stiffness I'm plotting here. That's the dimensional version. What we try to evaluate is what is the criteria for being in this tunable range. The criteria comes out to the overall fin length by that bending stretching parameter being somehow much greater than a parameter associated with anisotropy of individual rays. We can estimate that for mackerel fins, and now it's in the eye of the beholder whether 20 is much greater than 5 power 1/3 or not. I'll leave that to you. But at least it seems to be in this range where the mackerel fin lives. Its implications for the water to land transition and so on, and mud skippers we haven't yet looked at. So let me end by saying that what we found in feet work in fins despite differences in the internal structure, or possibly work in fins, but could work in slightly unusual ways where even a flat fin can behave like a curved fin. I think its implications are to really think about what we mean by flat or curved in these appendages, and the possibility of a common principle for these propulsive appendages, although their internal structure looks very different. And it's the principle which may be the thing that allowed the early evolutionary ancestor to move on land. I'm going to end with a slide just telling you what are the things that my lab does. You heard today about feet and fins. We now have money for coming up with designing a new institute that's going to think about appendages of different types on different terrain, with specific questions of how does morphogenesis lead to realization of related principles but with different morphologies, questions of evolution and morphogenesis. We've been working on problems in how does mechanics impact how neural control works in the context of throwing, in the context of fingers and how fingers are controlled, in the context of whole animal jumping. And somehow all of these in the back of our heads hinges on understanding how muscle can tune these responses. So there's a whole part of my lab and my brain that also thinks about muscle as a material and its rheology. Okay, I will stop here. I'm 5 minutes over time. And thanks to all of you for hanging around this one.
Thanks for the fantastic talk. That was very, very interesting. Does anyone have any questions?
So maybe I can ask a couple of things. Can you tell me again how you compute the curvature of your of the feet? And so I think it's one of the last slides towards the end. So my question relates to the fact that curvature technically is a pointwise measurement, right? But it can give informations to both global and local, and a little bit in between, structures, right? So that's why I'm asking, because maybe that slightly different ways in which compute curvature can give you slightly different information, right? Or even bigger information. So that's why, yeah.
So I'm estimating mean curvature. I'm not going to try to estimate the pointwise curvature. And mean curvature across the entire width of the foot for the reasons that you're talking about. And it's also then relevant that I use a consistent method for estimating these things. And that's why I'm here comparing. For example, I take the human foot or any primate foot, I can estimate the curvature associated with the envelope of the outside bones. That's just what you visually see and call as curvature. However, I can also estimate that using the torsion of the metatarsal because of this additional constraint that operates in the system, where the distal end of the foot is actually touching the ground, and therefore there are skeletal features associated with that. And that gives a coupling between curvature and the torsion in the foot, which we can show is a reliable coupling. And here I will add the caveat. I'm glad Armita is still here, because I'll point to Armita and say the kind of work she has been doing will totally alter how we want to even think about curvature. And the fish fin already shows that. Externally, it may look flat, but what I really care about is what is the soft bending axis associated with each of these long bones, and is the soft bending axis somehow systematically varying across the width? That we do not have an estimate for, because we really do not know how to go from shape to prediction of soft bending modes. That is an open question, I would say.
Thank you.
I had a quick question too. Most of the experiments that you were talking about here were sort of static in that you were, at least I think they were, you were hanging weights from the end of your either biological or mechanical models. And so, yeah, it comes back to this video exactly. So I guess I have a couple questions related to this. One is what the impact of the rate of applied force could be in affecting the deformation or the required stiffness. And then I guess also related to this running going is the directionality and of the change in stresses experienced by the different parts of the foot, because when you're running, you're not doing, most people at least aren't doing a heel strike when they're running. It's almost entirely, some people don't even make contact with their heels much when they run.
Yeah, so currently the recreational runner population, the vast majority are actually heel strikers. They are not mid-foot or forefoot. However, there is a large number who are mid-foot or forefoot strikers. That is also true. So in fact, let me add a third consideration. The foot itself is a living organ. It's not a static structure. It has musculature. We don't even understand how contraction of that muscle is going to change stiffness. But why am I thinking about static stiffness is a great question. Let me try to answer the simplest of the three. I can think about different deformation modes for the foot as a solid elastic body. The deformation modes are going to be quite complicated, but let me do a pretend orthogonal decomposition of a flexural mode, if this was the midfoot, a compression shear a compression mode, I would call that, not a shear necessarily, that's a tensile mode, and a torsional mode. I will for now disregard the torsion and tell you about the relative stiffness in the other two. The flexural mode is the softest. So we can ask what is the wave speed associated with the flexural mode in the foot, right? As you go along. That flexural, for the kind of cadaveric static stiffness we are measuring in the foot, that flexural mode is the wave would essentially go to one end of the foot and back in under a millisecond. The whole contact period, even in a running, for example, lasts about 100 milliseconds. So on that time scale, I think it is reasonable to now think of the flexural dynamics of the foot as flexural statics. The other modes are, and of course, therefore, the compression also is going to be static. But the torsional mode, I do not have even an estimate to base things on. So I do not know whether the loading rate is slow enough in running to pretend that it's quasi-static in torsion or not. You don't see any obvious oscillatory torsional waves, but then there's contact and dissipation, so I do not know how to interpret that. Muscle contraction was actually a worry, I would say, which is what prompted us to go look at what happens in living human subjects as they're walking and running. Can I even systematically change foot stiffness despite muscle contraction differences across people? I can, but and show consequences to foot energetics and stiffness in the middle of the stride. Beyond that, how exactly muscle contraction affects stiffness is a tricky question. There is a muscle group in the calf area which is close to the bone, which is called posterior tibialis. If in a person that muscle and the tendon beneath it gets affected, the arch just collapses. And it collapses because the posterior tibialis has a tendon which attaches to pretty much every single bone over here. So it's the bones by themselves are just a bag of marbles. They're not going to be held together unless they're a soft tissue, and muscles are part of the tunable soft tissue there. And we know some muscles affected. So there are others who are now trying to pursue this question in terms of how do different muscles affect the transverse arch because of its big role in static stiffness in dead feet. That's the motivating question. But we don't yet have data on. They don't yet have data to tell me how that's working.
And I know with muscle, there's also some groups that are starting to look at how like co-contraction of multiple muscles in an area changes the geometry of the muscle contraction. How like when a muscle bulges, it is pushing on everything else, and so there's constraints on the deformation of the muscle, and presumably in such a tight enclosed structure like a foot, with all of the fascia and tendons running through, there may be potential there as well.
Yeah, I would say the confinement of muscles within the foot is not too different from confinement in a lot of other places, because you have a bony constraint on one side, but the other is still softer and muscle under heavy contraction. Actually, maybe it's visible in one of the running videos. So I'm not giving away identities because this is me. On occasion, when someone's pushing off, you will actually, let's see, I'm going to try to track with. So look at the muscle that's in the middle of the longitudinal arch over here, and you will see that muscle bulge up very noticeably just when I'm pushing off. And it's not constant, and you will see that more in more athletic people than me. So there is merit to your statement about the confined bulging of muscle and interactions. Those are great questions. I don't have even inkling of how we can tackle these experimentally yet.
No, that's fantastic. Amrita has a question that she put in the chat. She asks, how do you have a sense of how the metatarsal 4 curvature proxy holds up across other primates?
Amit, a great question. I don't have enough samples in other primates. I'm mostly being saved by the fact that the other primates are pretty flat. And if you look at the metatarsal, so for 2 Aethiop samples, 2 Nemestrina, and I think 3 chimpanzees and 3 gorillas. Whether I use the 4th metatarsal or I use the external curvature, I get very similar numbers. This is the poor man's candidate before you can finish your understanding of how to infer soft bending modes, and that's what I don't know. You said, I wonder why. Yeah, absolutely. So I think there is some merit to the surface shape. It holds something about soft bending modes, but I don't know what.
Great. Well, thank you so
I've
much.
successfully chased everyone else away.
I suspect it was time constraints, possibly.
I think at one, probably people had,
going on to other classes or
I'm sure of that.
things. But yeah, if my technical difficulties hadn't delayed us, we would have been set a little bit longer, hopefully. But
we're gonna have follow-up questions for you, most likely.
absolutely.
Absolutely. I look forward to any chats and emails. I have fun with those.
Well, thank you so much for taking the time to speak with us. It was a very enjoyable talk. Yes. Thank you.
Thanks.
And I guess, yeah, we can stop the recording and yeah, thanks again. It was a fantastic seminar, and we look forward to
Stopping the recording now.
conversations.